our 5th session of Neuromatch 4. So we're in day 2, session 2, in case you've lost the plot already. And it's our final keynote talk. Things go fast. So I'm Jenny Bisley, for those of you who don't know me, and I am really delighted to be hosting Dr. Athena Akrami this evening, or this morning, depending on where you are. So Athena is a group leader at the Sainsbury Wellcome Centre for Neural Circuits, based here at UCL. Prior to that, she was a Howard Hughes Medical Institute Fellow at Princeton, working with Carlos Brody. And I think what's really outstanding about Athena's work is her ability to combine computational methods with behavior and systems neuroscience approaches for really kind of, engaging in this kind of iterative cycle of modeling and experiments. So I'm super excited to hear her talk. I'm going to hand the floor over to her. Her talk's titled The Formation and Update of Sensory Priors in Working Memory and Perceptual Decision-Making Tasks. Before I do, I would just remind you, if you want to ask a question, please use the Ask a Question tab. And I'm going to hand over to Athena. Welcome.
Thank you so much, Jennifer, and all the Neuromatch organizers. It's an honor to be here and talk to you tonight or this morning. Thank you so much for the intro too. So yeah, today I'm going to talk to you a little bit about, as Jenny said, it's kind of a cycle back and forth between experiments and model to try to understand something about how we basically kind of learn implicitly, and that kind of implicit learning can impact our working memory and further down the line, our decision-making processes. To start with, let me present to you some sort of a, hopefully kind of a fun clip to maybe a little bit wake you up. So I'm not seeing your faces. I hope this works. So I'm going to play for you a distorted sound. Just listen to this clip. Okay, probably you didn't really comprehend anything out of this. Now let's listen to this one.
The texture of experience is prior to everything else.
Okay, now let's go back to that distorted sound. Okay, so again, I'm not seeing your faces. I hope this worked, meaning that after presentation of the clear voice clip, you could then, going back to the distorted one, you could anchor yourself to some familiar patterns in order to denoise and disambiguate that distorted voice, right? So I presented this as a kind of a simple example of how basically kind of as we navigate in the environment, there are a lot of occasions that we deal with noisy and ambiguous situations, right? And we always use our prior knowledge, even if we are completely unaware of that, in order to kind of denoise these situations. So the question is that how we basically kind of acquire these big body of knowledge throughout our life, that most of it basically is unsupervised, right, and implicit, meaning that our brains, they evolutionarily speaking, they are evolved to be sensitive to regularities around us, right, that they are relevant to us. And most of these regularities, they unfold over time. And when the brains need to kind of learn these regularities over time, they basically kind of use our working memory in the back of our mind in order to piece together events, right? Like now that you are listening to me, and hopefully my speech is comprehensible for you, the reason is that even if you are unconscious about this, your working memory of different time scale is working in order to piece together for you the kind of words that I'm spitting out, from which you can infer my previous sentences, my previous paragraphs, and the gist of what I'm trying to say, right? And you may also wonder how babies, in the first place, they learn language, right? So use of language is something, and then learning of the language in the first place is something else, right? So babies, when they get to the age of around 7-8 months, they can kind of start to learn the syntactic structure underlying language, right? And meaning that they have their brain has the right hardware in order for them to start in a completely unsupervised manner, meaning without any kind of explicit instruction or explicit reward, they kind of learn the syntactic structure. What are the phonemes? Where are the word boundaries, and what is the relationship between the words, right? So the question that we are trying to kind of tackle in our lab is basically kind of how does the brain learn these complex but structured regularities that most of the time they kind of unfold over time, right? And we kind of know that there is we use our working memory, and here I'm really using the working memory in a very loose way. Maybe it kind of would make some of the human cognitive scientists angry, but by working memory, I mean some internal representation of our external world. And after that, we kind of extract something abstract out of this internal representation. And we use that abstract knowledge and kind of discard the rest of it, right? So this is what I mean by working memory. And our brain uses this working memory in order to kind of learn regularities. And the interesting thing is that once we learn regularities, they kind of impact our working memory down the line. So imagine that I asked you to remember a set of words. I think you all agree that remembering gibberish is way more difficult than remembering coherent sentence. Why is that? Because in remembering coherent sentence, again, we anchor ourselves via these learned regularities into some familiar old prior knowledge in order to kind of remember things, right? So in the lab, we try to kind of utilize rats, mice, human subjects, and computational models in order to study different kind of tasks that fall underlying this kind of this interaction between these two faculty of our mind, working memory and learning regularities. Some of them are very similar to the example of the language, which is just like abstract learning of temporal patterns in a completely implicit way. Some of them are related to kind of learning some variables that have some sort of a probabilistic distribution. For instance, like the learning the distribution of the height of the people around you, right? It's just like, it's not that we sit down and put down individual heights and we kind of measure the distribution in a very direct mathematical way. No, we just like by observing people around us, we kind of form some belief about the distribution of the height of people around us. How do we do that? How are we sensitive to these variables around us that have this probabilistic nature? And we try to really kind of study these type of behaviors related to learning regularities and learning prior knowledges in tasks that would allow us to basically kind of parse out different phases of the behavior. In tasks, we call them parametric. For instance, like parametric working memory, okay? And at first glance, they may appear very kind of unnatural, and I agree with that. We kind of a trade-off between trying to kind of make the task more controllable, right? We know that what are the different phases of the task, meaning that what is the sensory encoding, what is the memory maintenance, where is the decision-making part. And so as I mentioned, then we try to really kind of combine various levels, behavior in human and rodents, mice and rats, in high throughput basically system with various techniques to look into the brain and perturb different levels of neural dynamics in the brain, together with different types of models, either mechanistic kind of neural network model or more normative models, or dynamical system model, in order to kind of understand and build a multi-level understanding of inference and learning basically in this type of temporally extended tasks. Okay, so today I'm going to talk a little bit about this specific notion of parametric working memory and how we can kind of tap into some understanding of learning priors and use of that in the in-working memory. So, as I mentioned, we would like to have tasks that would give us this ability to disambiguate and deconfound sensory coding from memory maintenance, from decision making. And a very classic example of that is the work of Rodolfo Romo in monkeys, either in tactile or auditory domain, where, for instance, in the tactile, animals were receiving pulses, like trains of pulses on the fingertip. And these train of pulses, they had different frequencies, like, right, SA and SB. And they are separated by a certain delay interval. And the animal needed to basically compare the frequency of the second stimulus with the memory of the first one and then make a decision. Okay, this task, you may find it strange, but didn't exist until in 2014, together with Arash Fasehi and Matthew Diamond at CISA, we developed this task for the first time in tactile domain for rats. And then after I joined Carlos Brody at Princeton, I translated it into auditory domain. So it's basically a presentation of two stimuli, right? Two sequential stimuli. First stimulus, second stimulus, separated by a delay interval. And this delay interval can be as short as 100 milliseconds or as long as 20 milliseconds. And then while we are kind of basically looking into the brain or perturbing the brain during the memory, we are kind of sure that this whatever neural representation that correlates with the task, it's most likely correlates with the memory of the first stimulus, and it's not kind of confounded with the motor planning or future choice of the animal, because the animal has not yet received the second stimulus. Okay, so a kind of a closer presentation of the task in rodents. This is a top view, schematics of the top view of the task. So there is a operant conditioning chamber. The animal, it has like 3 pokes. The animal pokes in the center, receives a sound. This is like a kind of a noise sound, meaning that at each moment in time, amplitude of the sound comes from a Gaussian distribution. And by changing the variance of the Gaussian distribution, we basically are modulating the amplitude of the noise of the sound. So it is presented for 400 milliseconds. There is again a variable delay interval, second stimulus, and then go cue. And following the go cue, the animal can withdraw, go to the left or right side of the box in order to receive the reward. And the way that we are delivering reward depends on the relationship between the 2 stimuli. Okay. And we have basically equivalent tasks for humans, both in auditory and tactile domain. Okay, so let's now dig a little bit into kind of a nature of this type of working memory task. So as I said, we present subjects with the first stimulus that has a certain amplitude, right, SA. And then after a certain time, we present them with the second stimulus, okay? And what we ask them to basically, we expect subjects to compare the second stimulus with the memory of the first stimulus, right? But memory is inherently noisy. So our brains, the brain of this little guy, as well as our human subjects, can either kind of give up to this noisy representation of memory or somehow cheat in a smart way. Meaning that, imagine that the animal, after many exposure to different stimuli, builds some sort of a prototype sound. What is the most likely sound that can happen in this context? And then in this kind of a dimension that kind of represents loudness of the sound, when the actual sound falls somewhere here, and then if it kind of gets noisy, it may just like randomly move around and fade even. Instead of that, this sound may kind of contract toward this prototype sound, which can be built by looking at the recent history of the sounds. Okay, so what I am depicting here is a very kind of a Bayesian and maybe optimal strategy of dealing with noisy memory. Okay, meaning that we build this kind of a prior, what is the most typical sound, and we basically kind of snap our noisy memory into that in order to denoise it. Okay. And this, for the first time, was discovered by an American psychologist, Hollingworth. And I really recommend kind of reading his early works in various types of human psychophysics experiments. And particularly, he is the one who saved Coca-Cola for Americans, because when Coca-Cola came out, it had a really high content of caffeine, and regulatory agencies, they wanted to ban it because of this high level of caffeine. But then Coca-Cola kind of hired Hollingworth and asked him to, can you please kind of tell these people that there is nothing harmful in Coca-Cola? And Hollingworth ran really meticulous, very beautiful psychophysics, various types of measurements that would kind of show that, if anything, there is actually benefit in having caffeine. So, and he basically kind of discovered this phenomenon that it's been called various names. One of them is contraction bias. You can find it in under the name of assimilation to context or order effect, different names. But it basically means that the perceived value of items held in working memory, they contract toward the mean of the prior formed by all the stimuli being used in the task. And this has been since then replicated in various modalities in different tasks, mainly in humans. So if we go back to our task, we can kind of try to imagine what is the possible way of contraction bias showing itself in the task. So here I'm presenting again the stimulus set, first stimulus, second stimulus, diagonal means they are equal. And the color here shows the difficulty of the comparison task, right? So these two, like the memory of the first stimulus needs to be compared to the second stimulus. The closer to the diagonal, the more difficult is comparison. And here the vertical line shows basically the mean of the stimuli, okay? So under the assumption of contraction bias, if we have one pair of stimuli like these, the memory of the first stimulus will move toward the mean, toward this vertical line. And by virtue of that, that's going to get close to the diagonal, and the comparison task becomes more difficult. But then we do have other pairs, like this one here, that by moving basically the memory of the first stimulus toward the vertical line, it's going to go away from the diagonal, and the comparison task becomes easier. So then we can kind of divide all these stimulus set into different areas that we can call them bias plus, where contraction bias is kind of beneficial to the task, and bias minus, where contraction bias is actually kind of detrimental to the behavior, right? And we can particularly have some sort of a psychometric pairs of stimuli where we are manipulating the difficulty by changing the basically distance to the diagonal. And all of these psychometric pairs are now basically sitting in the bias minus region. That's kind of important because I will get back to that point later. Okay, so here is what we are presenting, the set of stimuli that we are presenting during each session to our animals. Or intermingled within one session. Also, there is, as I mentioned before, a variable delay interval. When we first look at the average performance over all pairs of stimuli for different delay intervals, one kind of things that, it seems that working memory behavior here is pretty stable from 2 seconds of delay interval to 12 seconds. But it's kind of cheating, and I will get back to that later, because it's very important to break down the performance for individual pairs and then look at some patterns that emerge, meaning that, look at here, the performance here is much worse than here, right? As we move this way, the performance decreases, and performance here is much worse than here. And we basically, as we move here, performance gets worse. And this is basically also very similar to what we found in the human subjects, right? Again, asymmetry of performance at the edges, and also as we move from a smaller value of SA to longer value of SA, the performance gets worse, and the other way around for second stimulus. And that's very similar, at least eyeballing on the data, to what we kind of qualitatively described for the effect of contraction bias, okay? But we can go beyond just like eyeballing on the data and find some kind of qualitative similarities. We can basically kind of pull out this impact of a mean of the prior stimuli, as well as other components in the task, by building a very simple regression model. Okay, so here the regression model assumes that the final choice of the animal is impacted by sensory inputs from the current trial, SA and SB of the current trial. We can have some bias term, but also some history impacts and parameters, meaning that sensory stimuli from one trial back, from 2 trial back. We can include also the reward history biases. This is something very well known, that like previous choices and previous reward status are going to basically impact the future choice of subjects, animals or humans, meaning that if someone chooses right and then gets rewarded on the right side, there is a higher chance of repeating the right choice again in the future trial. And then we can also include some sort of a long-term mean, which is basically a kind of a exponential window that integrates all the past stimuli and form this kind of a mean of previous stimuli, and then we can basically fit the exponential of that exponential window, the time constant of the exponential window, and find basically how different subjects are integrating the past. And the ideal performance on this task would have basically all the history terms, right? The weights for all the history regressors 0, and as well as the bias. The only non-zero terms would be the weights for the current stimuli, right? So equal with opposite sign. That's enough, and that's ideal to perform the comparison task. But nothing is ideal in real world. And when we compare here, basically these are your kind of, you don't really need to understand these plots completely. I'm here showing the coefficients for like the stimuli for the first, for the current trial, for rat subjects, human subjects. And the take-home message of that is, first, rat and human are pretty similar to each other when we look at different kind of weights for from this model fit. And then second, there is here we are showing the history signal, right? Weights from the history coefficients, meaning that the weights.
Nina, sorry to interrupt you. We can't see your mouse. I don't know whether you want to sort of change it to a laser pointer.
How can I do that? So
are you in PowerPoint or as a PDF or Keynote?
I am in PowerPoint.
I think if you hover in the left-hand corner, you should get a little menu, and you can right-click, usually to change it to a laser pointer, and then hopefully we'll be able to see it.
Okay. Do you see that now? No.
No, sorry.
I am sorry. Then I don't know how to do that.
I mean, you can probably just describe where you are. I just didn't want you to be thinking you were pointing at stuff.
Really sorry. Yeah.
Unless anyone out there knows. Full screen, really?
Yeah,
sorry, just seeing if anyone's got any ideas, but doesn't seem like it.
It is PowerPoint, and I don't see that option here. There is like, I can choose a pen, but that's different, but I can't really choose I don't know whether on Mac things are weirder. I'm so sorry. So I was basically pointing at, that's then difficult. At this left bottom plots, which are, I'm showing basically the weights for you see the x-axis says weight values, and then where there is a red box, it's basically showing the weights, significant weights from the model fit for the sensory for the stimuli from one trial back, for instance. And as you see, again, a lot of the subjects, they have significant non-zero values of the sensory history term, and human and rats are very similar to each other. And when we basically summarize, look at the summary of the sensory history versus reward history in this purple versus green plot in the middle down, you see that both of them are non-zero, but the sensory history part is basically kind of weighs larger in this behavior. Okay, another interesting thing, this plot on the lower right part is that, as I said, we can kind of fit different exponential windows with different time constants. And when we look at a tau for best fit for the individual rats, it seems that we kind of get a bimodal distribution of rats, meaning that some rats, it seems they are kind of integrating over hundreds of trials, and some of them are integrating over kind of tens of trials, which is very interesting. We'll try to kind of dig into that more now. Okay. So we have a behavior. It seems that we can show that in this working memory task, parametric working memory task, working memory is kind of clearly impacted by the history of sensory events as well as reward events in the task. So what can we kind of say about the brain? And so from the literature in primate and human, we already knew that like prefrontal and posterior parietal cortices are involved in working memory, but are they also involved in parametric working memory of graded sensory stimuli? That was not clear. And particularly in rodent studies, when you look at the kind of previous works, that they are not basically explicit working memory tasks, but they are perceptual tasks that rely on working memory, like delayed orientation or accumulation of evidence or multisensory integration. The result coming from PPC perturbation, PPC inactivation was very kind of confusing. Sometimes no result, sometimes impairment. So now I'm going to show you a set of results from optogenetic experiments and electrophysiological recordings in posterior parietal cortex of rodent brain that would kind of show and suggest that maybe PPC is one of the nodes, is involved in basically kind of integration of the memory with this prior sensory history. Okay, the first disclosure is that throughout the talk, I'm going to refer to this brain area as posterior parietal cortex. But the thing is that posterior parietal cortex in rat brain, very similar to all the other species, is a really large part of the brain. And by moving like anterior to posterior or medial to the lateral part, a lot of the things change, including the connectivity with the rest of the brain. So all my claims basically apply only to a very specific coordinate, as I'm showing in the basically in this kind of an anatomical plot in the middle, that applies to a very specific brain coordinate. Okay. And the first set of data that I'm showing you comes from full trial inactivations with optogenetics, meaning that we have animals injected with halorhodopsin. It's an opsin which basically has an inhibitory property, meaning that if that opsin expresses in that part of the brain and then later on we shine light, we will basically reduce the activity of neurons in those neurons that have halorhodopsin expressed. Okay. And full trial, meaning that the laser turns on as soon as the animal pokes in the center and hears the first stimulus, and then remains on throughout the trial until the animal makes a decision. Okay, so in the data plot in the bottom, I'm showing you that when we inactivated PPC, basically we managed to remove both bias plus and bias minus. So if you compare the yellow bars with the gray bars, you see that under laser on condition, basically the both biases, they go away, okay? And when we focus on these psychometric curves, now I'm pointing at these a plot of SA and SB on the top left corner, that you see the yellow psychometric pairs. And in that, you see all the pairs of stimuli, they are sitting on the bias minus region. And when we then look at the psychometric performance under laser on, the yellow versus control, you see that in 3 animals, this psychometric gets sharper, suggesting that the performance improves. This is something that didn't happen in the sham animals, okay? So, and that's again consistent with the idea that like by PPC, it seems it has these both kind of components of bias minus and bias plus, and when we kind of remove this bias minus, the performance improves. Okay, so, and when we fit the same regression model, again, the finding matches basically the behavior, meaning that fitting the model with different history parameters and comparing the control versus laser on trials, we found that the only regressors whose basically coefficient change, they are the sensory history regressors, okay? And the non-sensory history regressors, they remain the same, okay? So what happens in terms of the neural activity in PPC? You may wonder at this point that we should be able to kind of decode the sensory history that we were observing on the impacting behavior, and also kind of, it seems, kind of inactivating PPC is reducing that impact. We should be able to kind of decode that history signal in the parietal, posterior parietal neurons. So here I'm showing you one example neuron. X-axis is time, y-axis is firing rate, and you see that, and here we are plotting the firing rate in response to different levels of first stimulus, okay? So as you see, during the first stimulus, this neuron kind of fires the same no matter what is the sensory input. And it doesn't code the memory during the second of delay interval. During the second stimulus, also it doesn't show any kind of a modulation by the sensory input. And then go cue is played, the animal makes a choice, intertrial interval starts, and now suddenly from the firing rate of this neuron, we can basically decode what happened around 10 seconds ago, meaning that as you see, the firing rate grouped based on the SA that happened around 10 seconds ago, we can basically kind of decode the level of stimulus. This is very interesting because it's kind of a reactivation of these sensory events many seconds later on. So if now we go back at the kind of a summary of all neurons that we recorded in this task, which are like about 360 neurons over 5 animals, and we look at the mutual information between the firing rate of single neurons and different task parameters. So mutual information is basically kind of a tool that would tell us that if we do have 2 variables, X and Y, if we know X, how much our uncertainty about Y is reduced, okay? So we are looking at the firing rate, and we want to say that by knowing firing rate of a neuron, how much we can tell about, for instance, the stimulus identity of the current trial or the previous trial, okay? So everything non-significant is blanked out into blue in the heat map, and there is a population of neurons during the first stimulus that kind of codes the first sensory input. Then nothing of that really remains during the delay. Smaller population that again kind of keeps coding the first stimulus during the presentation of the second stimulus. Kind of silent during the ITI, and then a large population of neurons. Again, I'm pointing at the heat map that kind of starts having high and significant level of mutual information with the SA. Okay, so summarizing these heat map in the plot below, we are showing the percentage of cells with significant mutual information. Red shows the mutual information about current sensory input, SA, and blue shows the mutual information about the previous trial, okay, sensory input from the previous trial. And as you can see, when the next trial, this kind of a current trial starts, there is this population of blue here that these are basically neurons that keep coding still previous stimulus, not the current stimulus, okay? And if we kind of look at whether there is any kind of a correlation between this population here and behavior, we can simply, in the bottom plot, we can kind of first, over 5 different animals that we had, we can basically kind of sort the size of these, or the amount of mutual information carried by PPC neurons. And you see that there is like some animals, they have very small amounts of information, some animals have higher. And then we can go back to their behavior and ask how much in their behavior they are influenced by this past sensory input, right? And then there's kind of you get another sorting. And then there is a very clear, strong modulation between these neurons carrying sensory history and the amount of behavioral biases that the animals show. And so here we basically look at this neural activity during this delay interval of the current trial, but now we can basically move back. Here the x-axis shows different basically kind of a time epochs before the start of the trial. And look at the magenta line here. So magenta line, it's kind of peaks during the current delay, the delay interval of the current trial, right? Between like 0 to 2 seconds and then 2 to 4 seconds of the current trial, right? So, and when I went back and repeated basically the optogenetic experiments, but now focusing on different time epochs of the task, first stimulus, delay interval, or second stimulus, it seems that this impact of sharpening of the psychometric, consistent with removal of the bias minus in the task, happens if I perturb PPC during the delay interval. So it seems that kind of this perturbation study is basically consistent with this correlation study of the neuron and behavior, that like this correlation is picked during the delay interval, as if like somehow this history signal is integrated by the working memory sometime during the delay interval. Okay, so I have a summary here before moving to the kind of the next part, which is making sense of these sensory history biases via some modeling work. So first, working memory is impacted by history of sensory events. And this is something optimal given how noisy our memories are. And it seems that in rats, at least this specific coordinate in posterior parietal cortex is kind of causally related to these previous sensory history, and represents this part of the sensory history. And by removing this sensory history, it seems that behaviorally, we can detect the impact on reduced biases, the sensory history biases. Okay, so as I said, it seems that somehow this integration of the memory and this previous sensory prior history happens during the memory interval. Okay, and maybe some happens somewhere downstream of PPC. And why do I say that? I showed you some optogenetic experiments and also the neural correlate peaks during the delay. Also behaviorally, when we look at measure the bias plus and bias minus for different delay intervals, these biases increases, and they increase as we increase the delay interval, okay? And this is something that actually, when we look at another task that doesn't have the working memory component at all, so it's basically a sound categorization task. Rats or human subjects, they hear a sound and then they kind of need to categorize it into something above or below a kind of an arbitrary boundary. So in this task, that currently is being investigated by different people in the lab, Elena and Victor particularly, we cannot find contraction bias. There are other very interesting phenomena, meaning that subjects show interesting sensitivities to the underlying statistics of the sensory inputs, but not contraction bias. So it seems this kind of a working memory component is kind of important, and it kind of goes back to this notion of maybe optimal Bayesian framework, that we would like to form some sort of a prior knowledge, what is a typical sound, and we would like to somehow kind of, when we recall the memory, we estimate it through this prior, and that is a more basically optimal way of recalling noisy memories than just like giving up to the noise. Okay. On the other hand, I have to say that when we look into the behavior, I briefly mentioned that when I was telling you about the regression model, that there is a strong bias toward very immediate past trial, okay? Meaning that here, basically, I'm kind of quantifying that bias and like the heat maps, the x-axis shows current trial, the y-axis one trial back, okay, for our humans, rats, and in tactile domain or auditory domain. And you see there is a modulation by the y-axis, okay? Meaning that the memory stimuli in the current trial is impacted by immediate one trial back. Okay, so in the bottom plot, when we look at this amount of this bias, it seems that the bias from the most immediate trial is the strongest, and then it kind of dies, and then it's even stronger than this kind of a mean of a stimuli. Okay, so how then now we can reconcile the fact that there is these very short-term history biases toward immediate past trial, and then on the other hand, this kind of a Bayesian notion that contraction bias comes from this kind of a drift of the memory toward mean of the previous experiences. So in the remaining time, I'm going to briefly tell you some kind of a new insight that the work by Vijay in collaboration with Claudio Klopa's lab is giving us, which they were very surprising to me. And let me walk you through that. So Vijay is modeling this kind of interplay between the working memory and sensory history by having two basically continuous attractors. Okay, so we do have basically this kind of a bump attractors for the working memory network and also for the PPC network. And there is basically kind of a one-to-one mapping from the PPC network into working memory network. And the only difference between these two networks is that the integration time of PPC is much slower than working memory, right? That's a way for us to basically kind of model these sensory history. Okay, so PPC, both of them, they receive input, sensory input. PPC integrates it much slower, and then kind of sends these slow integrated sensory input to the working memory network. So if you look at how basically this network operates, at the time of the first stimulus presentation, okay, the working memory network, it's basically hovering somewhere random. Okay, this is before the stimulus 1. The stimulus 1 arrives, and then working memory network jumps into that. So it's a new bump representing the first stimulus. Then the stimulus 1 disappears. This is a kind of a attractor network. The bump stays around that. Okay. And then until the second stimulus arrives and it responds to the second stimulus. And the way that we are basically reading out this network is by taking out the activity at this time T1 and T2 to look at the difference between the two. So this is a case of a stable memory, meaning that the working memory is not really affected by the input from the PPC network. But we can imagine now the volatile memory of S1 in the working memory network. So here again, I'm showing you example of two stimuli. The first stimulus and S2 are very close to each other. S1 is smaller than S2. So now in this scenario, the working memory network jumps to the place of the first stimulus, but now it is kind of influenced by the input from PPC, and PPC input can kind of push this into a new place where PPC dictates where to go. And now this new location of the bump, right, where we kind of look at when we want to read out and make a decision, this new bump is going to give us a different result because this S hat of S hat 1, right, this new memory of the first stimulus is now actually larger than S2. So this is basically an example of bias minus. But we can also have bias plus situations, meaning that where PPC dictates and a working memory to bump to move to, now this new location is basically increases the distance between S2 and the memory of the first stimulus, and therefore it kind of increases the chances of correct answer. So it's producing bias plus. And when we look at the performance of our model on these different stimulus pairs and also this kind of a history heat map, you see that it's very similar to our real data, meaning that if you look at the plot on the left, When we move from smaller values of S1 to larger value, the performance decreases for the red boxes, and performance increases for the blue boxes. And that's basically very similar to the data, except for that, I'm sorry, the colors are switched. But it's basically kind of the same pattern, right? This asymmetry of the performance on this stimulus set, and very clear modulation by the previous trial, the identity of the previous trial, as you can see in the heat map blobs. The y-axis, there's a clear modulation by the y-axis, and that's the immediate previous trial. Okay, so what is exactly happening in this task? The thing is that, as I said, in some trials, memory bump survives. It's not volatile, and memory bump doesn't move around and gives us a kind of a faithful representation of the memory. But then in some other trials, the memory bump jumps to where sensory history input is, to where PPC tells it where to go, right? And mathematically speaking, at each trial, so we do have these pairs of a pair of stimuli, S1T, S2T at trial T, and there is 1 minus epsilon chance of basically non-volatile memory, meaning that S1T, actually this should be S hat 1 and S2 will give us the correct comparison, right? But then there is an epsilon chance that actually this new, this memory is volatile, and S hat is now moved to somewhere new. Where can it move? It can move either in a new place that would still give us the right classification, or move somewhere that now would give us the wrong classification, right? And where this S hat goes depends on basically distribution of the bump location in PPC network, okay? So if you have a kind of a uniform sampling of all these pairs of stimuli here, this is what we are going to get as a marginal distribution of stimuli, okay? And so this is the marginal distribution of stimuli. The distribution of pi, the bump location over PPC, is very similar to this, but it's kind of narrower because PPC is now kind of slowly integrating this. So if you kind of want to graphically present that, again, we have this stimulus set, and we are now showing just this kind of a distribution of bumps over pi, basically marginal bump location in PPC network. And this is the ideal performance, right? This is what we wanted to have. But now, if these stimulus is going to move, look at the darkest red box. Okay, so here it's darkest red box because if it moves, it's going to move somewhere on this distribution. And most of the places on this distribution, the ones that are on the right side of the diagonal, they are still going to give us the correct answer, right? So this kind of an influence of the PPC network or the history network is not going to have much of an impact on this one. But let's move up a little bit. As you see that now that we move, the possible wrong location, which means that the part of this distribution on the left side of the diagonal is just like increasing, right? So these pairs of stimuli, they have higher chance of moving somewhere, which is a wrong place for the comparison, right? Similarly, for this stimuli above the diagonal, right? As we move down, now we are basically increasing this red section, which means that the places that if this stimulus moves to, it's going to give us a wrong comparison. And that will just like, that is the reason that we are getting this kind of a gradual shift in the behavior in the average performance, okay? So summary 2 here, we show that our continuous attractor model for PPC, which is a slow integrator, can basically replicate almost all behavioral results that we had in our human and rat experiments. And this model suggests that this short-term history bias, which means that kind of a drift toward, it's basically just a kind of a jump toward the most recent past, can produce this seemingly contraction toward the mean effect that we observe it as on the kind of an average performance, right? So this contraction bias toward the mean is basically just an average effect over many trials, Whereas in each single trial, the volatile working memory jumps towards the input from the history network. And I think that was actually a very interesting surprise for me, because I really started thinking about this phenomenon of contraction bias under a very Bayesian framework. And I really would wanted to kind of go after finding some sort of a mechanism in the brain that would compute this kind of a Bayesian inference. But it turns out we don't need that really. Like mechanistically, we can have some operations that are completely non-Bayesian, but then on the level of behavior, basically they give us some sort of interesting properties that fit very well under the Bayesian framework, right? So it's just basically this kind of a confusion between, does brain do Bayesian computation explicitly, or basically we can basically model the brain function and the behavior and cognitive outputs in a Bayesian framework, right? So I can either stop here or tell you a little bit more about the next things that we are trying to kind of dig into in terms of the memory recall and the impact of the priors in the memory recall. Jenny, you told me I can totally stop here, or I can talk for like 4 or 5 more minutes.
I think there are quite a few questions. So if you'd like to, if we want to have a bit of a discussion, we should probably move to them. Although it's quite tantalizing.
I can just briefly, as a teaser, just let's say that. So in the working memory task that I talked to you about so far, the output is basically binary. So remember something, compare it to the second stimulus, tell me whether they are either similar or different, or larger or smaller, right? So it's just a kind of a binary comparison. And we are kind of throwing out a lot of information about the nature of the memory. So in other tasks that we are working on in the lab, we are focusing on a kind of a memory reconstruction task that would give us some continuous readout into the memory. And we are having it easily for humans, but Peter Vincent, PhD student in the lab, he's heroically basically translating this in rats and mice. But we also have some other special working memory tasks in rats that they would show this very interesting readout into the memory. So that's it. By that, I stop here and I thank all the members of LIMLab. And also all the collaborators that I do have at Gatsby, SWC, Imperial College. Thank you so much for your attention.
You get the sad clapping of one person. Thank you, Athena. That was beautiful. Okay, let's move to some questions. So one question, so I'm sort of going by the number of votes questions have. Can you scale the magnitude of the contraction bias with the variance of the stimuli?
That's a really great question. So I'm not sure whether I completely understand what they mean by the variance of a stimuli, but does it mean so? But definitely we can kind of change the variance of that noisy memory, right? For instance, by increasing delay interval. That's exactly what we were trying to do. When we increase the delay interval, the contraction bias clearly gets stronger. And our interpretation of that is that the memory becomes basically, if you do have some sort of a kind of a distribution over that memory, it becomes, the variance increases, it becomes noisier and therefore stronger, influenced by the by this prior, or under this kind of a mechanistic model that we are proposing, the memory becomes more volatile. That bump in the working memory network becomes more volatile, and it's therefore kind of easier for the PPC to move it around. So we definitely do have that. We have also some data when we are basically changing the distribution, how we are sampling the stimuli in this range. Now I showed you results from a uniform distribution, but we can totally change the distribution, and it seems, yes, that changes contraction bias as well.
Interesting. So a question from Anne Uri. Were the individual differences in rat behavior dominated by neural responses to sensory history, or could you also predict individual tendencies towards repeating choices or rewards?
Oh, fantastic question. Shame on me that I haven't looked at that. I do have the data, but I haven't looked at that. I should. That's a great question. The only thing that I can say that it seems perturbing PPC didn't really change the choice history or the reward history biases. Okay, even if I could totally decode choice history and reward history from PPC. Okay, so, and then later on, when in another data set, another behavior, I kind of looked in a kind of a comparative way, looked at sensory history versus choice history in PPC, FOF, and striatum. It seems that these sensory history and choice history are present on all these three networks, but the strongest sensory history is presented in PPC, and the strongest choice history was presented in FOF. So I say that, meaning I want to say that it seems kind of a choice. These are distributed. It's more about like which area might have a stronger influence, right? Which area might be more of a kind of a bottleneck in terms of downstream behavior. For sensory history, I'm pretty sure PPC is pretty important, but it seems for choice history, PPC is not as important as possibly other brain areas like FOF.
Interesting. So a question from Megan Peters. If the contraction bias is due to drift towards a prior, could you use dropout lines in a computational psychiatry approach to explore hypo-prior models, e.g. of autism and other failures in Bayesian inference in various psychiatric disorders?
That's a great question, Megan. Yes, yes. And actually, there are already some works into that. To be honest, so when I first really saw the result, my very first, I really didn't know about any of this when I started doing my inactivation experiments in PPC. I really wanted to go after just like maintenance mechanism underlying working memory, and I wanted to focus that. And then the very first set of data that I collected was sharpening of the psychometric, right? Which was mind-blowing for me. What is going on? There's something wrong. And the thing that kind of helped me a lot to understand what's going on and put together pieces of a puzzle was some work from dyslexia and autism related to contraction bias. And they are works mainly done by Mirav Ahisar. And she has basically a kind of a theory about like dyslexia, but now she's looking into autism. Now I'm collaborating with her, actually. We're looking into like autism, schizophrenia, and dyslexia. And it seems that like they do show different types of contraction biases. Types meaning that at the first glance, it seems like dyslexic people, they don't show contraction bias at all. And she has a theory that she calls it kind of an anchoring theory, that like dyslexic people cannot anchor themselves into predictions from the past because they can't basically build those predictions because they cannot integrate. Okay, this is the basically, and so she has done some works behaviorally before on these groups, and very recently there is actually a Nature Neuroscience paper together with Manish Sahani, comparison between neurotypicals, autistic and dyslexic in terms of time scale of contraction bias. So it seems both dyslexia and autistic, they show different way of integrating past compared to neurotypicals, meaning that one group, autistic group, weighs strongly the immediate past and not longer one, and autism is the other way around. So there has been now some kind of a definitely some interest in kind of understanding possible phenotypes in different psychiatric disorders that might point into different views of the sensory history, different views of priors.
That's super interesting. Okay, I'm going to have to close this session or I'm going to get in big trouble with the other organizers. Thank you again for a really beautiful talk, and also lots of food for thought there in the questions. Brilliant.
Thank you so much. Thanks, Jenny. Thanks, everyone. It was a pleasure.
All right. Hello, everybody. I hope that you enjoyed the last keynote. This was really a fantastic talk by Athena. And so now we are moving on to our next session of short talks. For those of you who don't know me, I'm Megan Peters. I'm a cognitive neuroscientist at the University of California, Irvine, just south of Los Angeles. And we have 3 wonderful short talks for you today. We have Maxwell Shine, Patricia Rubish and Nathan Close. And in that order, they are going to tell you about some really fantastic work that they have been doing. And so our first speaker is Max. Please take it away.
Awesome. Thank you. Great. So I'm going to talk today about some work that I did during my PhD with Jean-Marie Dale Lee and Hyojung Seo. And so it's going to address one question, which is, during perceptual decision-making, what happens when the evidence changes? So the problem with studying what happens when evidence changes is that right now, most of the task paradigms, most of the models that we use, most of the ways that we think about perceptual decision-making involve some constant stream of evidence that's roughly similar over time. And we don't really have the tools, the full set of tools yet to hash out what to do when evidence changes. How do we understand what happens when evidence changes? And what type of strategies do we use to account for these changes in evidence? So one of the limitations here is that we tend to think about perceptual decision-making oftentimes in terms of things like the drift diffusion model. Now, the drift diffusion model is a model where a model for 2 alternative forced choice tasks, where we have 2 alternatives represented by 2 bounds, one on the bottom and one on the top. And as we get evidence for one of the 2 alternatives, we tend to drift in the direction of that alternative. And we make the decision when this integrator type signal crosses the bound, when this decision variable crosses the bound. And one of the limitations of this is that it's only one single strategy that we can look at. It's one single strategy, and it really only accommodates a single type of task, which is a task that has evidence, which is, as you can see here, constant over time. So one of the things that I worked on is generalizing this so that we even have the possibility of studying tasks with evidence which changes over time. And so we generalize this to the generalized drift diffusion model. And the generalized drift diffusion model is very similar to the normal drift diffusion model, which you can see here, except for it has several extensions that allow us to try and understand different strategies that one might use during perceptual decision-making. So for instance, we can do bounds that change over time. We can do evidence that changes. So our drift rate can go up and down in whatever way we want. It can depend on time. It can depend on the position of a particle. Position dependence allows us to do things like leaky integration. We can have changes in starting point. We can do several different generalizations of the standard drift diffusion model, which in the end allows us to test different strategies. But not only does it allow us to test different strategies, but it also allows us to look at different experimental paradigms. So for instance, if we have evidence that goes up and down over time, maybe evidence that kind of spikes like this, maybe some Poisson-like type of task, maybe we have evidence that's constant with a pulse somewhere. These are all things that we can do very easily with this generalized drift diffusion model framework. And if you're interested in looking in this more, we have a software package called PyDDM that you can look at. You can also look at our eLife paper, which is linked at the bottom of this slide. So now that we have the tools to study different strategies, the tools to understand different task paradigms, we can go ahead and start looking at what happens when evidence actually does change. And to do this, we're going to use this type of a task in monkeys. So how this task works is, first of all, I want you to look right here where I'm pointing. And the goal here is to try and determine whether there are more blue pixels or green pixels in this square patch. If there are more blue pixels, then the monkey is supposed to saccade to this blue target. And if there are more green pixels, they saccade to this target in order to respond. However, what makes this task different and what allows us to study evidence which changes over time is that we don't start out by showing the monkey this stimulus all the time. Sometimes we'll show the monkey this stimulus right here, which I'm going to call the pre-sample. The pre-sample is identical to the sample, except that the ratio of blue to green is 50-50. So there's no correct answer here. And if the monkey responds during this time, then we give it a timeout punishment. So it's encouraged not to respond during this time, respond during the sample period instead. But importantly, there's going to be a change here from when we go to this pre-sample period where there's no stimulus to this sample period where there is a stimulus. Let me show you a quick movie so that you can get an idea for this. So first of all, I'm going to show you an easy trial and then a more difficult trial. And so the easy trial, which is where it changes to a high level of color coherence afterwards. So there's the pre-sample, and then it swaps. And you can see that was an easy trial because there's lots of blue there. But they're not always that easy. Sometimes we have hard ones with a lower coherence. So maybe you could see that one change. I couldn't. The monkeys can do better than chance. The monkeys are better than I am at this. But this is kind of the task that we use here. So if we look at the response time distribution of this, we see something like this. So let me walk you through this. So the colors here represent different durations of this pre-sample period. So this orange is no pre-sample at all, and this red is 800 millisecond pre-sample. And then the brightness of these different colors represents whether it was an easy trial or high coherence, which is the bright colors, or whether it was a hard trial where it looks more gray. And this is kind of hard to interpret. And what we actually care about is this period, like right here and right here, right when the evidence changes. So let's zoom in right here. But before we zoom in, let's say, what might we expect to see here? So if you think about pretty much every model that's been constructed of decision-making, we're going to expect to see something like this, which is we're going along, there are some RTs here, and then suddenly when this changes, we might expect a ton more responses when we have the easy trials, because once your evidence changes, now the task is easy, you're going to probably respond very quickly. Versus if it changed and you couldn't tell it changed, you're going to respond somewhat similarly. And so this is what we expect to see, and this is a simulation provided by a generalized diffusion model. What we actually see is this. Now this is strange. Let's walk through this for a second. So this gray is when there's no change. So this is when it's 50% of each color throughout, and that kind of goes up like this. But when we look at these really strong trials where we would expect a ton more responses, because this red line is where it changed, right? So this is where it changed to become really, really easy, but we get a reduction in responses for this short period of time. After this, of course, it goes way up and you have lots of responses after that. But during this short period of time, we have a reduction in the number of RTs on the easiest trials when it changes to something easy. That's really weird. I had no idea what was going on when I saw this. So next step, we looked at behavior, did some neural recordings from this. We did some recordings from the frontal eye fields. The frontal eye fields are nice for this type of a task because it contains sensory information, it contains motor information, and it also contains information about the decision-making process. So sensory information because each of the cells have a receptive field. It shows a ramping-like activity during evidence integration. And it also is able to generate saccades if you stimulate. So this is maybe a good place to look for this task. And now we can look at frontal eye field activity. And this is exactly what you saw before, except for instead of RTs on the y-axis, we're looking at FEF activity on the y-axis. And let's zoom in again right here. And again, what we might expect from any model that I've ever seen of this is that it's going to keep going up. And then it's going to keep going up over time. And then once the evidence comes on after some efferent afferent type delay, there's going to be a huge increase in frontal eye field activity, and for the easiest trials and not for the hard trials, sort of like you see here. And this is directly from a simulation. But what we see again is this exact same effect. We have a reduction in frontal eye field activity when we have stronger evidence compared to when we have weaker evidence. And this is this does end up being graded too. So the stronger the evidence, the bigger the reduction for this short 100 millisecond to 200 millisecond period. This is very, very confusing. And we're talking in the panel discussion earlier about how models can help us make sense of something when we have no idea what's going on. This looks like a perfect time to do some modeling here. And one way to do modeling is to think about evidence integration in 3 steps. So the first step being evidence comes in, and then there's some decision variable that does the integration. Maybe it does fancy stuff, maybe it's leaky, maybe it's something like that. Some type of decision variable that does that. And then we have motor output, where it actually makes the saccade to respond. And we'll think about this change detection as the monkey having a probability of detecting the change. So on the easy trials, it's very easy to detect the change. And on the hard trials, it maybe detects it with a lower probability. I'm going to show you the high probability ones here, just for simplicity. And so if we detect a change, let's suppose that it inhibits the incoming evidence. The intuition for this might be, oh, well, the evidence is volatile. We want to wait until it stabilizes, so then we can have something constant, and decision-making can be like we like it with no evidence changes. But when we do this and simulate what we would expect for frontal eye field activity based on the decision variable, we don't get this dip that we got before. We don't get this reduction after the stimulus comes on right here. Instead, it's just flat, and then it goes up, it goes down. This is very similar to what we would expect from traditional models. Okay, what about this? This is maybe makes the most sense, possibly from a normative perspective, is if you detect a change, then you take your decision variable and say, I was halfway through this task. I thought I was integrating evidence. I guess I wasn't. Here's the real stimulus. I'm going to reset my decision variable back to 0 and pretend like I'm starting from square 1. This made a lot of sense to me when I started looking into this. And when you look at it, it does produce this type of a dip, except it only produces it when the frontal eye field cell that you're looking at is within the receptive field. When it's outside the receptive field, it doesn't produce that dip. So we have one step left. So we can test this. We haven't shown whether this is true or not. Let's see what this would be different if it were inhibiting motor output. So in this way, if you detect a change probabilistically, you're instead of touching the decision variable stays exactly as it is. You just briefly pause your motor output. If you briefly pause your motor output, you see this dip, but you not only see it within the receptive field in gray, you also see it in cells outside the receptive field that you can see in red. And this is because there's this rebound effect right here. And when we look at what's actually inside the frontal eye fields, we see this rebound right here. So this is giving us evidence from the frontal eye fields that this motor suppression model is maybe a pretty good model. We can also look at what happens when we're fitting these models, since these are all generalized drift diffusion models, so we can very easily fit them to data and evaluate their log likelihood. And we have the strongest model coming from the motor suppression model. So this model where we're inhibiting motor output. So it's great for predicting frontal eye field activity. It's also great at behavior. Is there a test that we can maybe give it? Maybe some type of irrelevant motor activity? I know one. How about microsaccades? So in theory, microsaccades should be completely disconnected from the decision variable. The decision variable should not influence microsaccades. And when we look at this, we actually see there is a reduction in activity here too. So motor suppression is occurring even for types of eye movements that are not relevant for the task. So this is giving us pretty good evidence that what's happening here is that during decision making, when there's a change in evidence, when there's a sudden change of evidence, there's a brief suppression of motor output. And from a normative perspective, you might think of this brief suppression of motor output as something like you're integrating and your integrator keeps going, but your integrator is slower than this suppression of motor output. And so you want to suppress motor output for a second to give the integrator time to catch up with what you see. And so thank you to my 2 advisors during my PhD, who were John Murray and Dale Lee. Hyojung Seo collected this data. and Norman Lamb contributed to the generalized drift diffusion models. So thanks a lot, and I'd be happy to take any questions.
I will clap for everybody. Thank you very much for the really engaging and interesting talk. We have one question from Conrad, actually. So Conrad asks, to which degree should we think of the DDM models as mechanism or how models that are supposed to predict neural activities versus as normative or why models that maybe just predict behavior?
Yes, I think that's a very good point. I think that we can, I mean, it depends on how you want to interpret it and what context you want to interpret it. To a certain extent, we know that the DDM cannot possibly be neural activity. Neural activity cannot go below 0. You can say, okay, well, maybe there's some nonlinear transform that we can apply, and maybe this nonlinear transform somehow maps onto the neural activity. Then you start getting complicated. That's really hard to figure out. I think that there's some room for a middle ground of not necessarily saying this is exactly what's going on in the brain. These are exactly the variables. Can be, this is optimal behavior. This is what we'd expect. However, in addition to being descriptive, we can say, okay, well, if this is happening, we might expect to see some type of a signal like this. And so there are, so for instance, there are other things that could cause the rebound. It could be a local circuit property. It could be tons of other different things. But if we're saying that this is due to, this type of mechanism, then we can't have that mechanism, like a reset mechanism, for instance. We can't have a reset mechanism alone explaining this rebound in frontal eye field activity. So either we can say it goes to something else or it goes here.
Awesome, thank you. We have one more quick question, which is, was there only impact on reaction time, or was accuracy also impacted?
Yes, that's a great point. So if we go back to the response time distribution. So actually, this is another interesting point, is right after the evidence changes, right around this period of time, there's a short increase in error responses. And it's a transient increase in error responses. It's more than you would expect just from the sudden increase. And it's like right around here. And so what this is suggesting is that this motor inhibition in some sense failed. And that's a very good point. We have actually a different paper on this in Journal of Neuroscience 2020. If you're interested more in that error response, we go into that a lot more in that paper.
Fantastic, thank you. Well, I have questions too, but I think in the interest of time, we need to stop and move on to our next speaker. So maybe we can move this conversation to the Discord channels. Okay, great. So thank you very much. That was a really engaging and interesting talk. And our next speaker is Patricia Rubish. And Patricia, the stage is yours.
Thank you so much. Okay. Okay, my slides are up. That's good. So, yeah, thank you for having me and for giving me the opportunity to talk about my most recent work with Matthias Hennig. And what I want to do today is to introduce to you a novel pathway-based plasticity model. And by doing that, I also show the validity of the model and how it reproduces some standard experimental data from Synaptic Neuroscience. And we'll show you a few predictions of the influence of variable subthresholds membrane potential dynamics, which you can normally find in biological neurons. Okay, to start, I want to give you a quick overview of the voltage-dependent pathway model, or VDP in short. I will just use VDP to save a bit of time in the rest of the talk, and point out the very novel aspects of this model in comparison to conventional phenomenological models. As the name itself says, this model is a voltage-dependent model, and it focuses on postsynaptic plasticity-inducing processes. It is modeled as analogous to the NMDA-dependent long-term potentiation pathway. So this is the pathway shown in the flow diagram here in yellow. And the depression side of this model is modeled in analogous to the mGluR mitigated LTD pathway. So if you follow the blue parts of the diagram, this corresponds to all the depression-inducing variables. An important aspect of the VDP is that all plasticity updates are modulated by a postsynaptic calcium signal. So in short, this means if there is no calcium, there won't be any plasticity updates. The time courses of these three main components and the presynaptic component, the glutamate signal, are also plotted as the time development traces here on the bottom. I hope you can see my mouse, but if you follow my mouse or just look at the bottom plot of this, you can see the traces and the development of the activity over time of these four main components, which are important to compute the overall weight change. The processes here are actually induced by a positive spike pair, which means that the presynaptic spike is followed by postsynaptic activity. While the VDP emphasizes dynamical nature of plasticity-inducing processes and modeling those as by exponential differential equations, it does make this more complex than conventional voltage-dependent models, but the model itself does not reach the complexity of detailed biophysical models. So in fact, this means that the VDP is computationally efficient and can be used for large network simulations. Therefore, you could say that the VDP actually builds a middle ground between complex biophysical and efficient phenomenological plasticity models. And just to pinpoint this really, really clearly, the main difference between conventional phenomenological models, disregarding whether they are based on calcium or voltage, is the order of the thresholding operations. So usually in these kind of plasticity models, you do have at some point in the equations a step where you take the variable you want to look at, so the membrane potential or the calcium levels, and threshold them. Just compare them to a set threshold and say, okay, now it's a positive or a negative change. In the VDP, so in conventional models, this is actually one of the last operations done in the model, and directly influences what sign the synaptic change has. In the VDP, however, which is also a soft-bound threshold model, this thresholding operation only sets off the dynamics of the LTP and LTD pathways, and therefore only indirectly decides or influences the polarity of the weight change. So as you can see here on the bottom right plot, is that with most of the spike pairs, you will always have a phase where the synapse undergoes first potentiation and then gets depressed. It's just the difference between the overall potentiation and depression which decides what weight change the synapse undergoes in total. And to carry on, I want to show you the results of the replication of central experiments from Synaptic Neuroscience. So first, as it's a voltage-dependent model, it should be a given that the relationship between postsynaptic membrane potential and the weight change can be replicated. As you can see here in figure A, this is actually the case. The curve follows really nicely the effect often described in literature. So you do have a phase where the synapse undergoes depression first, and then at intermediate membrane potential values, you have a reversal point where after that, with increasing potential, you will observe potentiation in the synapse. Okay, then as a second nice part of the VDP is that you can manipulate the dynamics of the different pathways and therefore decide whether you have a fast LTP activation or a slow LTP activation, for example. By manipulating these dynamics and changing the temporal constants and their amplitudes, the VDP can replicate different STDP kernels described in the literature. The setup here, just to mention this really quickly, is that we have a presynaptic spike train, which is connected to a postsynaptic neuron, which we actually simulated as GLF5 neuron. And these two are connected through VDP synapses. to have closely timed postsynaptic spikes, we injected a very high current into the postsynaptic neuron in one single simulation type step. So in this case, it would be 0.1 milliseconds, which forced the neuron to spike in the next time step. So back to the kernels. I have to admit, the first kernel, which is shown here, so the symmetric LTP-LTD kernel, is a bit of an outlier. It is an outlier in the sense that it needs a slow glutamate signal, which is not the case for all the other kernel types, and it is highly variable to the parameters used. So even a very slight difference in the parameters can completely destroy the whole kernel in itself and revert it to either Hebbian or an anti-Hebbian STDP kernel. But to give you a better impression of what actually I mean by changing the dynamics and what kind of kernels need what kind of dynamics, I want to characterize the last 4 kernel types, which are a bit more stable with regard to the parameter settings. So the Hebbian STDP kernel is, for example, characterized by fast LTD pathway and a slow LTD pathway. And in contrast, the anti-Hebbian STDP kernel needs exactly the reverse setup. So this one will have a slow a slow potentiation pathway and a fast depression pathway. For the symmetric single polarity kernels, those can have different dynamics of the pathways, but they are mostly characterized by the dominance of the respective amplitudes, which means that the potentiation pathway is way stronger for the symmetric LTP kernel than the depression pathway amplitude, and the other way around for the symmetric LTD kernel. The results I've shown you so far are focusing on only isolated spike pairs. So if you look at an example for multi-spike pairing directions, for example, the experiments here shown in C, we see that also the behavior of multi-spike pair interactions, often described in literature, is reproduced. The experiments we performed here was actually a replication of the protocol for the pairing frequency effect reported or presented by Systrom et al. In 2001 for the first time. For this protocol, we repeated 5 either positive spike pairs or negative spike pairs at varying frequencies. And you can see the frequency here plotted on the x-axis. So positive spike pairs, so a pre-spike followed by a postsynaptic spike, shows a positive proportional relationship with a pairing frequency, which is described very often in literature, while a negative spike pairs first induces depression in the synapses until a reversal frequency, which lies between 20 and 30 Hz normally, and then induces potentiation as an input frequency effect takes over, which normally is the stronger the frequency, the more potentiation the synapse undergoes. So these were replications. There is another experiment you can perform to see how multi-spike interactions actually works in different plasticity models. However, there is not a lot of data to compare these triplet simulations to, but I wanted to show you the results anyway. So what we have done here is that we had the same setup, but instead of spike pairs, we paired triplets of spike in all the possible configurations between pre and postsynaptic spiking. And the 2 heat maps here show all the different configurations where one of the quadrant has the corresponding configuration of spiking plotted on top of it. So you can see directly to which combination the results actually refer to. So in general, the VDP predicts a very strong effect of postsynaptic spikes and exact postsynaptic spike timing, actually, which is not surprising if you keep in mind that the model is mainly based on postsynaptic induced plasticity processes. The results also show a nonlinear effect between the 2 different interspike intervals within the triplet, which is not a novel aspect found, but it's in so far contradicting as it does not agree with the hypothesis that one of the two interspike intervals is more decisive of the sign of the synaptic change than the others, which is a common assumption done. But again, unfortunately, there's not a lot of data to compare this to, so this is a prediction rather than actually a feature, a replication. The results I've shown so far are based on only one of the possible 15 excitatory neuron types of the GLF5 neuron model. And before I go on and discuss the predictions for all of these neuron models, I just want to reiterate about why we use the GLF5 neuron model and why it is so important that actually the plasticity model is compatible with this neuron model. So the GLF5 model is a neuron model including after spike current, voltage-dependent reset, and a variable spiking threshold. And especially the voltage-dependent reset impairs conventional phenomenological voltage models from deriving consistent predictions for different subthreshold behavior seen in biological neurons. Furthermore, the GLF5 model has been fitted to replicate electrophysiological behavior of cortical neurons in mice, as presented by the paper introducing the model in 2018. And a clustering of those fittings resulted in 15 different neuron types, which behavior is actually here shown for 4 of these neuron types. And you can see that the behavior after the spike generations is distinct within all of these neuron types presented. And this is exactly what is impairing conventional neuron models, conventional plasticity models to derive at consistent predictions for all of these 15 neuron types. However, the VDP is actually able to predict stable results and especially stable SCDP kernels for all 15 neuron types using the same parameter settings in all of the experiments. But if you compare the relative area of potentiation to the relative area of depression, so the LTP to LTD balance of these produced neuron types, you can see a group difference. And interestingly, this group difference is between conventionally excitatory labeled neurons, which would be the group here from E0 to CTGF, and conventionally inhibitory labeled neurons. So the neurons from E9 to the parvalbumin neuron. The excitatory neuron group does show a few outliers, but these outliers can be directly referred back to the subthreshold dynamics, and they show a very big difference in their behavior in the electrophysiological properties above, as you can see with the example of the CTGF neuron, which is the second plot in figure A. Interestingly, this group difference is not present if you look at the results for the pairing frequency protocols. The pairing frequency protocol actually illustrates the influence of individual dynamics of the different neuron types. While the majority, so overall 11 out of the 50 neuron types, reproduced the reported effect as shown before, and here again plotted with the example of the neuron type E2, you can find qualitatively 2 different variations of this behavior. The first one is shown by the example here of E0, and you can see that both positive and negative spike pairings are actually affected with these variations. Both positive and negative spike pairings show a decrease in amplitude of their update, while the sign stays the same. The second variation is only affecting the positive spike pairs. And here you can see that there is a modulation at intermediate spiking frequencies. So instead of increasing consistently with increasing pairing frequency, for these neuron types, you actually see a decrease in the amplitude for positive spike pairs at intermediate frequencies, and then an increase again for larger frequencies. Both of these variations can actually be argued to result in either self-regulations of the synapse and the synaptic change, or input frequency sensitivity but further experiments are needed to investigate a possible functional component of this behavior. And this is actually just the results of the parent frequencies experiment. And with this, I actually come to conclude my talk already. I just want to reiterate the main takeaways and advantages of the VDP again. So the voltage-dependent pathway model is an efficient, biologically grounded model. It therefore offers a compromise between the advantages of phenomenological and biophysical models, and its validity is established by the replication of central experimental results, of which I showed you a subset. A further advantage of the VDP is actually its compatibility with more complex neuron models, which allows for a study of the influence of electrophysiological properties in standard plasticity protocols. Such a comparison predicts a strong variation of LTP and LTD balance in SCDP experiments and a strong nonlinear influence of dynamical components. The source of these components can either be the subthreshold dynamics of the neuron types used or of the pathway, voltage-dependent pathway model itself. And to give you a short outlook, we actually plan and already are in the middle of expanding the setup to network experiments investigating the functional role of different SCDP kernels, the neuron types and subthreshold fluctuations induced by inhibition in a more complex network setup. And with this, I want to thank you for listening and shout out and give a special thanks to my supervisor, Matthias Henning, and the Neurons and System groups, who gave me encouragement and very valuable comments throughout this work.
Thank you very much. Really interesting talk. Unfortunately, we do have to move directly on to the next talk. But if you do have questions, please put them in the Ask a Question box, or we can go to Discord for some more discussion. So thanks. And we will now go to our last talk of the session. Nathan Close, are you ready? Go ahead. The stage is yours.
Yeah, so let me share my screen. All right, so normally you should sit. Okay, perfect. So I'm going to present you some of the work that I'm doing as part of an internship in the lab of Robert Yang at MIT. And this will be about the modulatory role of the prefrontal cortex when solving simple cognitive tasks. So recurrent neural networks have been used as a way to understand how the dynamics of a population of neurons can give rise to some useful computations to solve some tasks. And as an example, I will consider these results by Mente et al. From 2013, where they recorded the activity of the prefrontal cortex of monkeys doing a contextual decision-making task. So briefly, in this task, we have two different sensory modalities. We have the motion of the dots and the color of the dots. And depending on the contextual cue, it is only one of the two sensory modality that is relevant for the current trial. So if we analyze the recorded activity of the neurons, we notice that it is actually low dimensional, and we can visualize it using some targeted dimensionality reduction methods. I'm not going to detail this now, so if you want, you can go to and check the original paper to have more details. But what is important is that when we train recurrent neural networks on the same task, we also observe these low-dimensional dynamics. And so these trained RNNs have been used as a mechanistic hypothesis about how the dynamics of the neurons in the PFC are actually solving the task. So far, so good. But then my point is that when we are doing this, we are actually assuming that it is the prefrontal cortex that is implementing directly the computations. But if we take a larger picture and consider the fact that the prefrontal cortex is actually embedded inside a network of cortical areas, the function of this PFC might be different. So let me explain what I mean by that. So here we have assumed that the function, the role of the PFC is more transmissive. And by that, so here I have a small diagram to illustrate what I mean by that. So we have this the prefrontal cortex that is represented by the box on the top, and we have different cortical areas that are depicted below. And so some of these areas are receiving the sensor inputs and others are producing the motor outputs. And in the case where the PFC has a transmissive role, it is directly computing the task response from the sensor information that it gets from the modules, from the sensory areas. And then it transits back the response to other areas so that they can produce the correct behavior. Okay? But this does not really capture what we know about the modulatory role of the prefrontal cortex. So in this case, the role of the prefrontal cortex is more to orchestrate and to organize the communication between the modules, which are themselves implementing and doing the work. So, and this idea is really not new. For example, there is this paper by Miller and Cohen from 2001 that argues that the role of the PFC is more modulatory. And so, in order to take into account the possibility of having a modulatory role for the prefrontal cortex, we have developed an RNN, a recurrent neural network architecture, that models the prefrontal cortex as part of a larger network of cortical areas. Okay, so I'm now going to describe this RNN architecture that we have developed. So in this architecture, we have what we call a cognitive system. So that's a simple recurrent neural network that models the PFC, and it has access to the contextual cues of the task. We then have a modular RNN that models the network of cortical areas. Some of the modules are receiving the sensory inputs and others are producing the motor outputs. And there are some interactions between each module of the modular RNN and the cognitive system. So there is also something special in this modular RNN, because unlike traditional RNNs, modular RNN, sorry, here the communication between the modules is implemented using an attention mechanism, which is inspired by this machine learning paper by Goyal et al. From 2019. And the motivation between this is to model the dynamic reconfigurability of the functional connectivity that we observe in the brain. And so thanks to the attention mechanism, the connectivity patterns between the modules can dynamically reconfigure itself. And we will see later that in some models, the cognitive system influences this dynamic routing between the modules so that it depends on the context. All right, so we now have this RNN model, and we can train it on a bunch of tasks. For example, we trained it on several reaching and decision-making tasks. And we can then try to analyze and understand what is the function of the cognitive system in this model. Okay, so the first analysis that we did is to look at how the task performance are impacted if we prevent the cognitive system from accessing the sensory information. So we silenced the communication from the modules to the cognitive system and look at how it impacts the task performance. Okay, so typically what we observe is that the performance will be greatly impaired. And so the model where the communication from the modules to the cognitive system is silenced, these models cannot solve the tasks anymore, which suggests that in these models, the cognitive system has a more transmissive role. So the cognitive system is responsible for computing the response of the task and then transmitting back to the output module so that it can output the correct response. And so when it doesn't have access anymore to the sensory information, the whole model cannot solve the tasks anymore. Okay, and so how can we have models where the prefrontal cortex has a more modulatory role? And so to have that, what we did is to add some constraint to this interface. And so I'm not giving all the details, but the idea is that we had a bit of a delay to this interface. And in that case, now the whole model has an advantage to use the modules to solve the tasks. And we found that using some constraint, now the model is able to solve the tasks without having the cognitive system to provide the correct solution directly to the modules. Because in this case, the cognitive system can only provide the contextual cue to which it has access. Okay, so this suggests that in some models, we have a modulatory role for the cognitive system. But to investigate that a bit more, we can look at the communication patterns between the modules and how it evolves during a trial. Okay, so now we will consider the contextual decision-making task that I described briefly on the first slide. And so in this task, we have three types of sensory inputs. We have a fixation cue that indicates when the model can output its response. And we have two sensory modalities, where the first sensory modality is the relevant one in the first context, and the second sensory modality is relevant in the second context. Okay? And we can now analyze the attention mechanism that is used for the communication between the modules in order to understand how the routing between the modules evolved over time. Okay, so here I have represented the query and the keys of the modules. So if you're not familiar with the attention mechanism, the idea is that the motor module here will output a query, and each sensory module has its own key. And the motor module will be attracted to the sensory module which has the key which is the most similar to the query of the motor module. So for example, during the fixation period, so at the very beginning of the trial, the query is here the dotted arrow in black. That's the query of the motor module. And we see that it is the closest to the key of the first sensory module. That's why the communication at the beginning of the trial is from the first sensory module to the motor module. And as the trial evolves, the query of the motor modules will move, and the keys also. And during the decision period, near the end of the trial, now the motor module is attracted to the second sensory module. And so here I've shown the attention weights for the motor module. And we see that at the beginning it is attracted to the first and then to the second sensory module in the first context. But in the other context, the motor module is attracted to the second sensory modality, which is corresponds to the third sensory module. And so here is the full picture. Depending on the context, the routing will change once there is the co-cue. And here it is really the influence of the cognitive system that makes this dynamic routing context dependent. Because if we remove the influence of the cognitive system, then we don't observe this anymore. So here is an illustration of how we can use this attention mechanism to understand how the routing and the communication between the modules evolve in a contextual dependent way. It was really on a simple task, but we can also do it on more difficult tasks, and also with more modules to have more complex routing. But yeah, this will be for some future work. So now, yeah, it's near the time of the presentation. So I will conclude. So I've presented an RNN architecture that takes into account the fact that the prefrontal cortex is inside a network of cortical areas, and that it can have either a transmissive or a modulatory role. And we have found that depending on the level, the amount of constraints that we add to the interface between the modules and the PFC, the role that it will take will change. And so some of our future work now involves looking in more details into the population responses in the cognitive system of our models, and try to see if we can observe similar population responses than the one observed in the brain. And we found that already in some of the models, even though the cognitive system has a modulatory role, we can observe similar population trajectories, which might suggest that the modulatory role is compatible with what we observe in the brain. All right, so this ends my presentation. I'm now ready to answer any questions you might have. Thanks.
I think I clap for everybody in the audience. Thank you very much for this really interesting presentation. Do we have any questions from the audience? If not, maybe I have a very perhaps naive question, which is this modulatory role that that you've kind of built in this. I'm not familiar with the attractor dynamic kinds of plots that you showed on the previous slide with these keys, but would the same thing happen if you had the context show up later after the sensory information has been presented? So you don't know which thing you have to pay attention to ahead of time, because it feels like the motor preparation is, maybe this is a naive question here, but does that make sense? That does it matter when you present which type of motor response or which type of sensory information you're going to have to attend to?
So if I go back to this part, we see that really at the beginning, before that the stimulus are shown, the two keys of the two sensory modules that contains the sensory modalities are really, they coincide, so they are on the same line. And this is really the information about the context that will differentiate them. So that will make the one corresponding to the relevant input go towards the query of the motor module.
So it doesn't matter when the query is presented, whether it's kind of at the same time as any sensory information or afterwards. The contextual modulation that's driven by PFC could happen kind of any time in that chain. I see.
Yeah.
Great. Thank you for clarifying. I appreciate that. So if do we have any other questions at all? All right. Well, it looks like we're a little over time anyway for the whole session, but that's okay because we had an engaging set of 3 talks. So I want to thank all 3 of our speakers again for their really excellent presentations, and to remind everybody that we also have the Discord server for offline conversations if you have any questions that you didn't get answered here. And go and have a quick bio break, and we'll be back here in about 8 minutes for our next session of short talks. So we'll see you then. Thanks.
Good evening. Welcome to Short Talk Session 7. Sorry for the couple of minutes delay. We're just trying to connect one of our speakers who will hopefully join us for her slot at the end. Really excited about this group of talks. I'm going to hand more or less straight over to Laureline. Do you want to start sharing your screen?
So thank you very much. I'm going to share. And I'm going to discuss a theory for the population rate of adapting neurons that allow us to understand how single neuron properties shape neural population statistics. And really what I'm going to discuss first is that there is a little bit of a rift in how people model the neural response today, whether they think about isolated neurons in slice, which are spiking, reliable, and modeled with more detail, or whether they're trying to capture the response of networks in vivo, where they often rely on rate equations with tuned connectivity. So the reason why it's possible to go with more details in slice is that it's possible to patch single neurons and to really control the current that is injected in the neuron and to record the voltage. And what you observe is that the single neurons are going to respond with these large spikes that occur at relatively reliable times with millisecond precision when you inject the same current. And it's possible to model this really well using models that decide spike times as a function of the voltage and the time of previous spikes. But when thinking about neural networks in vivo, in contrast, when using these rate equations, what people will often do is to develop, for instance, functional models where you have a network, a rate network, and you train it to return a particular output, for instance, an electromyogram if you're modeling, say, motor cortex. And you can compare trial average firing data with the model. Another way that people use rate network is for empirical descriptive models. You want a network that is going to feed the data, and this is a pipeline that will produce firing rates from which spikes that are, as you can see here, very asynchronous, very noisy, are drawn. And the question really is, those red squiggles, those firing rates in those rate networks, how do they mechanistically relate to single neurons dynamics? And it's a question that has been studied in the past using theory, but there are some limitations, notably to take into account adaptation, and that's what I'm going to talk about today. And the reason why I'm going to talk about adaptation is that if you really want to have a neuron model that is going to capture the response of a patched single neuron, the way you model it is you're going to have, at least in some models, a probability of spike in a very small time being that is going to be proportional to a very small intrinsic noise, which is a very small probability that the neuron would spike in the absence of input, and a nonlinear function, here an exponential, that includes two different terms, including a filtering of the input current. And this filtering is a filtering through the membrane filter K. And this gives us a drive to this neuron such that after passing through the exponential nonlinearity, some spikes are being drawn. And what the spikes are doing also is that they are filtered through this adaptation kernel here, an adaptation filter. And these spikes, S here, what they do is at each spike time, when they get convolved by this adaptation filter, they decrease the probability of spiking of the neuron. So what this gives you is this very reliable spiking response. And if you knew what the current looks like in vivo, in principle, you should be able to understand how this single neuron then responds in vivo and how that could relate to rates. So the question really is, what is the input current in vivo, where we don't inject it, but it comes from the network? And a clue, because it's hard to measure those input currents directly, comes from already looking at the firing from simultaneously recorded neurons. And as I'm showing here, this firing in vivo, it tends to be very asynchronous. And on top of that, even though there seems to be some correlations at the level of the rate of these different neurons, once you condition the response on the population rate, there is very little correlations between the spiking of two different simultaneously recorded neurons. It's very close to a random shuffle that would have no correlation at all. So this is the asynchronous state. You have this uncorrelated spiking across neurons, often in vivo, and that also happens in recurrent circuit. Modeling suggests that this is due to excitation inhibition balance, and that also implies that the total excitatory inhibitory current response when a population of neuron is stimulated externally is approximately uncorrelated across neurons. So uncorrelatedness in vivo is really what should be taken out from this slide. So then the question is, can we think of the rate as an average activity of a population of neuron? And what are the statistics of the input in this population in vivo? And what that means is that we have this population of neuron with different pyramidal neurons, I and I+1. There is also fast inhibitory interneurons in this population. Here we're going to take them so fast that the response is going to be assumed to be almost instantaneous. And we have different populations that interact with one another. And we want to try to understand the population rate in the black population here. And as I mentioned, really what that means is that we also have to try to understand what the input looks like in vivo. So we have these different groups of neurons, G, that project to each neuron of this population. And so the input is going to be the sum over the different groups of the synaptic currents that are being driven by spike trains from neurons in the presynaptic groups. And these synaptic currents, you can get them by convolution by the membrane filters that we talked about before, and this effective synaptic filter, which in our case is really this already balanced excitatory inhibitory response. And the sum of all of these currents invoked by the presynaptic spikes are the drive. And the question is, what is this drive looking like in the asynchronous state across different neurons? And I told you that there is some reason to believe that this drive is relatively uncorrelated between neurons. Mathematically, what that means is that if you have different presynaptic groups, K and H, with population rates RK and RH, which projects through these different neurons I and I+1, the expectation of the product of the currents that are evoked by these presynaptic spikes is more or less equals to the product of the expectations, which is themselves just depend on the rate. The consequence of that is that the drive D is this sum of many, many weakly correlated and identically distributed variables for each group. So it's approximately Gaussian across the population, and it has no time-dependent mean and variance. And what we want to try to know is what is the population rate that averages over the single neuron statistics over this distribution of drive in different neurons of this black group. And so mathematically what that means is we want to compute the expectation of this firing probability lambda. And in our models, there are two types of stochasticity for this firing probability. There is this intrinsic noise, which is very small, but which gives a little bit of variability in the response for a fixed drive. And then there is expectation over the drive itself, which changes across neurons. And for the first expectation, previous work has shown that you can have a very good approximation on timescales that at least are longer than the very short refractory period by having a slightly different adaptation kernel eta tilde that now convolves not the spike train, but the private spiking probability of the neuron. And all we need to do now is to compute the expectation over the different drives, which may not be as simple as one might hope. One can do a first naive attempt to compute this expectation by neglecting the variance of the drive across the population. This would be a naive rate type of model where you're just replacing the drive and the adaptation by their averages. And then you can compare what happens in simulations and in the model for different means of the drive and different variants of the drive that are different shades of gray and shades of cyan in the theory. And what happens is that the theory not only it strongly underestimates the rates because of this, it neglects this exponential nonlinearity, but also it's qualitatively wrong in the sense that different variances of the drive now give you the same response in the theory, whereas in the data, the firing rate increases when you increase the variance. A second attempt that has been suggested in the past is to just neglect the variance of the adaptation across the population. So the adaptation is the same as we had before, and now we account for the exponential nonlinearity, and we have a contribution of both the mean, the expected drive, and the variance of this drive. This assumes that the adaptation is much slower than all the other timescales, but this is not accurate given realistic adaptation timescales. Really what this approximation is saying is that you have these different neurons, I and I+1, they have these different drives, they have these different adaptation shown in brown that correlate with their private drive, and you're trying to replace these specific brown drives with this average purple curve that's the same across neurons. And this is not going to work very well, as you can show here. Can see here, the firing rate is overestimated. And here you really have to compare the black curve with the dark gray curve, the gray curve with the medium green, and you can see that they don't match. So the third attempt, which is our contribution here, is to consider adaptation as a linear filter of the drive. What that means is that we can replace this neuron-specific firing probability here approximately by a linear filter, that gamma, that we can compute analytically of the drive D. And so what that means is that now the rate is the exponential of a convolution of this drive D, which is itself Gaussian. So it's an exponential of a Gaussian variable. The prediction of this already would be that the instantaneous rate distributions should be log normal. And this is indeed what we often see in vivo. And we can now look at what happens in simulations and in this equation here. And now we're getting a very good match across a larger range of mean and variance of the drive. The other thing that we can do is to look at the response as a function of time in a non-stationary regime. And you can see that this also works well. The red and the black are pretty close to one another, and a naive rate equation does pretty bad. Finally, the last thing that we can do is to actually express and work out this expectation here. And what we are going to find is that this gives us a response that has a mean-driven regime that's the same as the naive mean-driven rate model in attempt 1. And the time scale of this mean-driven response is relatively slow. But it also has a fast variant-driven response that accounts for both the drive and the adaptation. And these relative timescales, they don't depend on the specific parameters. And so they make prediction for the timescale of response that you can actually verify in the data. You can patch neurons or a single neuron and record its average response when you change the mean at a particular time of the input versus when you change the variance of the input at a particular time. And you can see that when you change the mean, you have both a slower increase of your response and also a slower adaptation. So this works relatively well. This is consistent with what we see in slices and in vivo. And what this means is that we were able to replace this big population of neuron with a rate equation here. And something that you can also work out is that this rate equation here is really equivalent to a system of coupled nonlinear differential equations, which looks like your typical rate models, except that now you have several variables and several different timescales for each population. So it's a higher dimensional rate model, a little bit like an LSTM in machine learning. And so what we have seen is that really, with this approach, we can understand how rates emerge when you're considering a network of population of neurons, and each of these population of neurons has a higher dimensional rate equations. And this also paves the way for asking whether these biologically derived higher dimensional rate equations may be beneficial when trying to train networks for specific tasks. And with this, I would like to thank you for your attention.
Thank you for that. It was really interesting and really clear. I'm afraid for timing reasons, we should probably move on, but people should feel free to either ask you a question with the tab or on Discord. You can go there and kind of continue conversations.
Thank you.
So Catherine, do you want to start sharing your screen? Next up, we have Catherine Shapcott, who is talking about rank similarity filters on computationally efficient machine learning on high dimensional data.
Okay, I think this is working now.
Yeah,
Hi,
that's good.
I'm Catherine. I'm working in Frankfurt in Germany, and yes, I will talk about rank similarity filters. So that's a novel type of artificial neural network that's very efficient. You can check the preprint in the agenda if you're interested, and also the code is all on GitHub if you'd like to try it out. So yes, similarity judgments are something that the brain can do very intuitively. So all of us can hopefully do the following task. So if I show you what is probably a novel object for you that you've never seen before, you'll already be able to tell me how similar it is to other objects. So for example, this bike and this car, you can see in which ways it's similar and in which ways it's different. And this is what we call one-shot learning. So already, just by seeing this object once, still not really knowing what it is, you can already make these similarity judgments about it. So how might the brain do this? So I think this could have something to do with vector quantization. So this is a method of data compression. So instead of needing to store all the data of all the bikes and all the cars that you've ever seen, you can just store a few prototypes of the data. And these prototypes can then be used for classification because they contain information about similar data. So similar data gets grouped into one prototype. So for example, here, maybe these would be the bikes and these would be the cars, right? And the prototype bike could be this in the middle, prototype car. And your novel object here, because it's well outside the other groups of data, could be its own prototype, and you can already decide how similar or different it is to the other groups of data. And you can also see in a high dimensional space which features are similar and which are different for this. Okay, so this is the theory kind of, but how could neural networks do this? So actually, it's very easy for them to do this. So, right, a node in the neural network would be, it has weights, right? So the vector of weights, and these weights could be its location in the space. If you would then have input, the neuron that have weights that matches the data better. So in that case, the blue neuron here, it will have a larger response. the red neuron here would have a smaller response, and they could inhibit each other. And then this would be the prototype for this new data. And then the weights could update so that this would become the prototype of this class. And you could have as many prototypes as your network allows. But the problem with this is that firing rates are fluctuating all the time. And we're not mapping a stationary space as in the previous slide, but actually we know that the firing rates are changing constantly. So how can we handle this? So what we thought was that actually we might use the confusability of features. So this is a new measure. Basically, what it is that you have here, for example, this blue feature. This is very far away from all the other features, and it's very different between the classes. So this would be a feature that's not very confusable with the others, and it would be highly weighted. Whereas this green and yellow feature, they're much more similar to each other, much more likely to overlap, and then you give them less weight. But what you maintain is the ordering of the features. And especially when you get to very high dimensional data, which is what the brain is good at dealing with because each neuron tends to have tens of thousands of inputs, actually the specific values in this high dimensional space don't tend to be as meaningful anymore, whereas the order of the inputs are meaningful, even in very high dimensions. So this we maintain. So here you can see in a fashion MNIST dataset, which is commonly used, that was on the first slide, that the rank follows very nicely. Can someone turn off their mic? Then you see that the confusion is following the rank here. So in this case, for this data set, you don't even need to calculate the confusion. You can just use the order statistics, so the rank of the data, and this already works for weight setting. In another data set, 20 news groups, this is text data. This follows an exponential function. Here, the rank is no longer a very good approximation of this, and you do need the confusion to set the weights properly to deal with this data. So I decided to make an algorithm of this. So because it was such a simple idea, and it's very, very fast. So all you have to do is create this distribution of confusion if necessary. You initialize the neurons or the filters in the space. You spread them through the space based on this rule of the winner then takes the data and updates its weight accordingly, and then you normalize them. And then you have your prototypes already. And the spreading here is just done by a dot product of the neuron with the data and then the winning neurons or prototypes. They are the ones who then update using this rank of the mean of all of the points that they won. And you do this until the points don't change their prototypes anymore, which can sometimes just be as few as 5 iterations, maybe up to 20. And yeah, so it's extremely fast. So here would be an example prototype vector on the fashion MNIST dataset. And what you would do is multiply it with a new piece of data that comes in. So in this case, a sandal. You do the dot product, and this is the response. And so here are the winning prototypes in this case. And you see that they're all similar looking sandals. And you can use this to transform the data set. So here you see just some random example data fed in, and these would be the top responding filters. So again, because it's quite high dimensional, the absolute distances don't matter as much anymore. So we just keep the top responding ones and their relative responses, and transform the space this way. And you can set all the rest to 0 because it's not meaningful anymore. So if we take a support vector machine and we apply this rank similarity transform to the data, so this is the raw score of the SVM, so it's linear, so it doesn't do so well in this data set. If we give just 26 of these prototypes, it's not very good. But as soon as we move to 150 of them, you can see that it's beating the raw SVM. And by the time you get to 1,500, it's really doing quite a lot better. And the really cool thing is it's also a lot faster. So with 1,500 of these prototypes, you see that it's still only taking 10 seconds. And this includes the time of the transform and the SVM running. So it really is quite a lot faster. And that's actually increasing the dimensionality of the data. The original data is only 784. So yeah, that's a pretty nice result. And actually you can directly use this as a classifier. So that's the rank similarity classifier. So here you see actually the performance is even better. And here we have the number of samples. So we're making the task here. It's quite difficult. And already you see very good performance with just 1,000 samples or so. So this is also nice. And it's even faster again, because now we don't have the time of the SVM, but instead we use the labels of the prototypes to assign a class to the data. Yes, and we can compare this. There are some other classifiers that have been tested on this data set. And so the parameters, the best parameters that could be found with a parameter search on the Fashion MNIST data set. And this is the rank similarity classifier here. So you see that it's not reaching the performance of, like, for example, an SVM with a radial basis function here, but it's really an order of magnitude faster. So that's like a huge difference in speed for quite a small performance gain. And this is after a parameter search, so it's even slower to find actually all of the correct parameters that you need to get such a good performance on a nonlinear data set. So yeah, that was it. So what I would like to do next is test the biological plausibility of this. So what I'm trying to do at the moment is implement the algorithm on neuromorphic hardware. And I would also like to try and use this as a weight setting method for a recurrent network because it's so fast that this could be a very good starting point for then further training to take place. And I'll be doing recordings in monkeys in the Havnet and Scholfink lab in a very dynamic task where I should be able to see if we see this type of maintenance of ordering, because we'll have quite some number of channels during the task. Yeah, and I'd like to thank Alex Bird, who helped me a lot with this project. Yeah, and the entire Singer Lab and the Instrument Institute and the FIAS for support. Thank you.
Thank you. I shall clap on behalf of everyone. You stuck very nicely to time, so we do have time for a question while people may be composing their thoughts. Is there any reason why your approach wouldn't work for like sounds or multisensory stimuli? Or, I mean, obviously you've been doing visual images.
Yeah. No, I mean, it works on text data, for example, right? What it's not very good at, because it's usually orders, if your features have no relation to each other whatsoever, then the ordering becomes meaningless. So this it cannot do. So I don't know, there's some data sets that, yeah, it just doesn't seem to understand them. I guess if you would get the correct scaling, because it's very rare that actually that the different features have nothing to do with each other. It's quite, you know, they're always somehow related. But yeah, if they're very highly unrelated, then it won't work at all. And the other thing, it's not good at low dimensional data. So you need enough dimensions to have enough ordering to actually get enough information out of this. But that in the current day and age is not really a problem. We have plenty of dimensions of our data.
Yeah. Well, and in the real world too, right? I mean, the real world has many dimensions. It doesn't look like there are any more questions, but again, people can follow up either here or in Discord later. Dory, I'm really pleased to see you're here. Oh, there is a question. Let's look at the question. While Dory maybe shares her screen. So would this algorithm handle translational invariance in images? That's from Carl Johnson.
No, so it's single layer. So no, it can't handle that. That's another thing I would like to work on. If I can layer this, then it should be able to do that. But then I need to section the other layers, and I haven't found a good way to do that. But yeah, it's just because it's a single layer, it can't handle that.
Cool. Thank you again for a lovely talk. Dory, are you there? If you're there, you're muted.
There we go. Can you hear me?
Yes, we can.
Excellent.
Share your slides?
I am trying to.
Okay.
Okay. I do apologize. I had to run to grab someone else's laptop, but it's an Apple and I'm not used to this. So I should be able to share screen. Let's see.
So Dory's going to talk to us about a novel population of synchronously active pyramidal cells in hippocampal area CA1 when her Apple laptop plays ball.
Can you see anything?
No.
No. Well, that's disappointing.
I mean, I have your slides emailed to me, so I can let me download them. And worst case scenario, we can do it kind of in an old-fashioned next slide, please, kind of.
Yes. It just, yeah, I don't know. I'm clicking share and it doesn't do anything.
Shall I share them?
Yeah, if you could do that, that would be great. Thank you so much.
So let me. Go with that. And then
Excellent.
You see it? Cool.
Yeah. Great.
So I don't see you now, so you'll have to tell me.
I will. Okay. So sorry, everyone. Thanks for bearing with me on this. I will hopefully now talk about my research that I did at the University of Sussex. I'm currently in UCSD in San Diego, and also just moved here, so I don't have Wi-Fi yet, so hence all the issues as well. But we're here. So what I want to talk about is this population of synchronously active pyramidal cells that we found in hippocampal CA1. Next slide, please. So what I was doing basically is I was working on another project, and we had these Taiwan GCAM6F mice. So with the Taiwan, meaning that they're fluorescence in the excitatory cells. And we were doing the surgery, a hippocampal surgery, to insert a cannula over CA1. And what this does is it basically gives us a window just over CA1 where we can do 2-photon imaging on these pyramidal cells in hippocampus. Next slide, please. So we had our mice in this virtual reality. So you can see it sort of, this is a 3D rendering from the back. You can see the mouse on the ball, and it can just move as it pleases, or it can stand still if it wants to. And as it moves, this virtual reality will move around it. And you can see the sort of lens there as well, which we use for the 2-photon imaging. So I was doing this experiment and sort of sitting near the microscope and looking at my cells. Next slide, please. And I noticed these sort of very synchronous, weird cells during the recording. So that should be a video. I hope you can play it. If you can't, it should be okay, but
No, it looks like
that's fine. So we saw these highly synchronous cells. So believe me, they are very, very cool. I'll post a link in the Discord later on. Interestingly, we only saw these during stationary periods, so when the mouse was not moving, and the activity seems to be periodic. And so for these two reasons, I'll call them stationary low frequency oscillatory cells, and the rest of this presentation are slow cells. So we don't have the video, but I do on the next slide have a little graphic to show you sort of their behavior. So what you can see at the top here is 6 cells, the 6 that the arrows were pointing in the last slide. And you can see that the delta F over F here. So the more green is higher activity, basically. So you can see that these 6 cells during this sort of short rest period, it's about 3 minutes, just over 3 minutes, that they are active basically all at the same time every time. And you can see at the bottom as well, when you look at the average, which is the black trace is the average activity over these 6 cells, you can see that there seems to be some sort of periodicity to it as well. So they fire at a relatively sort of regular interval. So next slide, please. So I wanted to try to, I put that link on the wrong slide. Anyway, I wanted to try to see what percent of cells this was at first. So we took only 8 data sets because we couldn't see them in all data sets. And across these 8 data sets, there are about 6% of cells, so very small population. But you need to take into account that this was 8 data sets out of tens of data sets. So it's probably in reality an even smaller populations of cells that we see here. So my first step was to try to characterize them. So next slide, please. And first I wanted to look at their activities. As I said, by eye, it looked like they increased their activity during rest. So quantifying this, we showed that the activity between slow and the control cells, which is basically everything that's not the slow cells, is very similar during locomotion in green, but the slow cells increase their activity during rest, whereas the control cells decrease their activity during rest. So they have this a very sort of particular pattern of activation, essentially. So this was the first question I wanted to look at, which was the stationary sort of a characteristic. The next thing was the periodicity. So if you could go to the next slide. So used a Welch's method FFT basically to look at the power frequency spectrum, the power spectrum. And you can see here the slow cells on the left and the control cells on the right are plotted on the same y-axis. So you can see that the slow cells have a much, much higher activity or much, much higher power in this low frequency. So the x-axis only ranges from 0 to 1. When I control for the number of cells, because the slow cells have a much lower number, which creates like more noise in the average, when I controlled for that with the control cells as well, this particular band of interest between 0.1 and 0.4 hertz emerged. So here we could see that basically there's this band of interest where these cells particularly fire. You can see a similar band also emerge when we look at the difference between rest and locomotion. Next slide, please. So a question, of course, that emerged because these are cells in CA1 is what are they doing in terms of place? Because CA1, famous for its place cells, wanted to look at the sort of overlap between these cells and the place cells. And found that there was to some extent, an overlap. So you get about 10% place cells, whereas in the control cells, a bit more, so about just over 20%, which is sort of what we would expect. Do note, though, that, as I said, it's a very low percentage of cells. So we're talking about 5% of total cells are these slow cells. Then 10% of them are place cells. So in a population of about 200 cells, this is one cell. So that makes it a bit hard to say anything conclusive about it, but there definitely seems to be some overlap. Next slide, please. So then what I wanted to do is see if they actually had a function in spatial encoding. So first, looking during locomotion, when obviously the place cells are firing, we see that the place cells in pink have a higher spatial information than the non-place cells in blue, which is expected. But interestingly, there's no difference between the slow and the control cells. So they have the same sort of pattern of spatial encoding. However, so next one, please. If we look during rest, we can see that this pattern actually changes a bit. So we can see that suddenly the slow cells, so again, the left side, so the non-place cells in particular, but also still the place cells, have a very high spatial information during rest, higher than the control cells. So what we think this might indicate is that the slow cells maintain some information about space when the mouse is resting. And this fits with some, there's not a lot of literature on these kinds of cells out here. That's why I call it a novel population. But with similar sort of research has found that this to be the case. But obviously, this is not very conclusive evidence. So this is something that we're still wanting to work on. Next slide, please. So just wanted to give the overall takeaway. So we have this novel population of highly synchronous cells, which we call the slow cells, which have increased activity during rest compared to the control cells. And they're particularly active in this frequency band of 0.1 to 0.4 hertz. And they seem to play a role in location encoding, particularly during rest. Next slide, please. So that leaves me to thank the Hall Lab, as well as Caswell Barry and Roman Bourbelou, who have helped loads on this project. And also I need to thank Jenny for all your patience with all my technical issues today. And thank you for doing the slideshow for me today. I'm happy to answer questions here or over Discord.
Cool. Thank you very much. And well done for kind of keeping your cool when everything is. Let me... No, let's not close the video. How do I stop screen sharing? Okay, we'll just have infinite screens for a moment. There are some questions. Now mirrored infinitely. So how exactly did you pull out these slow cells from the rest of the population? Was it a criterion on oscillation frequency or synchronization at rest?
That's a very good question. So this is something that this has been basically my main sort of issue with this whole project is it's very hard to, it's so easy to see them and it's very hard to sort of pull them out. So what I ended up doing with this is using this, what you said, the synchronization at rest. So basically I sort of did a recurrent algorithm where I add in the next most synchronous cell recurrently until I see a decrease in sort of the overall synchrony of this population of cells, and that seems to work. So basically, there's a natural sort of dip when I do this, and that seems to work really well in pulling out exactly this population.
So another question: can you explain what is the frequency power measure that you use? Is it the coherence of these spikes with different bands of the LFP?
No, so the frequency was, because it's calcium imaging, I can just take like this straight up the calcium data and just perform the fast Fourier transform on that. So we did also look at LFP, and there doesn't seem to be actually a very strong sort of coherence between the LFP and these particular cells. So this is in a different data set. We had LFP and calcium at the same time. But we think this may be due to just the low number of cells. So it seems like there may not be enough cells of them to really influence the LFP. Or another reason could be that the LFP is localized. It seems to be quite localized to certain areas of CA1. So if we don't have the probe exactly in that area, it's hard to measure it.
So I was going to ask you, and you're sort of touching on it now. You said that you found these slow cells in 8 out of 10s of animals. Was there anything like, could you pinpoint differences in the region you were targeting, or the mouse's experience, or like anything that would allow you to figure out why those animals and not others?
So I haven't actually looked at the experience yet. I do need to do that, but it seems to be particularly in some animals and not in others. So I think out of 12 animals, it was only in 3. I don't have, in my original data, I don't have enough information about the exact location, basically because we weren't doing this project to look at these cells. But I have now looked at the Allen Institute data. So they have some great data out there with some neuropixels as well. And using that, it does seem to be very localized to sort of more ventral CA1.
Interesting. Cool. Well, if there are no more questions, which I don't think there are, I will thank all 3 of you again for really lovely talks. You get the sad solo clapping. And remind you all that you have a little over an hour now in which you can either enjoy your virtual meetup or go to the Flash Talks or follow on discussions on Discord. So thank you for attending this session, and we'll see you back in a little over an hour.